Hi, my name is Lauren Van Woudenberg, and I'm presenting on a new genetic abnormality that could potentially be used in the development of a male contraceptive. Dr. Richard Smith of the University of Iowa published these findings in the April 2nd edition of the American Journal of Human Genetics. Female oral contraceptives have been developed for over 40 years and have become extremely commonplace in today's society. However, no similar pharmacological contraceptive has been developed for males. CATSPER1 is a gene that is involved in non-syndromic male infertility in humans. This means that the infertility is not part of a syndrome. This could lead to future infertility therapies that replace the gene or the protein. The research team collaborated with scientists from the University of Social Welfare and Rehabilitation Sciences in Tehran. They discovered the male infertility gene while studying genetics from families in Iran. Iranians have a relatively high rate of disease-causing gene mutations, which makes them really interesting to study. This particular study noted 2 families where male infertility was not part of a syndrome and appeared to be inherited. From there, researchers focused on a group of genes that has been implicated in male infertility in mice. They were able to find that these mutations in both Iranian families occurred in a single gene called CATSPER1. Harvard University did further studies on mice that lacked the CATSPER1 gene to reveal how it affected sperm when the protein was missing or abnormal. These studies found that CATSPER1 mutations affected sperm motility, or the ability to move spontaneously and actively, consuming energy in the process. Specifically, they noticed that the very vigorous hyperactive motion that sperm use when entering the egg during fertilization was compromised. Research suggests that the defect in sperm hyperactivity that is seen in mice without the CATSPER1 gene will also occur in humans with the gene mutation. What the CATSPER1 gene method is an immunocontraceptive. An immunocontraceptive is a birth control method that uses the body's immune response to prevent pregnancy. One of its popular present uses is to control wildlife populations, and it's designed to provide long-term, not permanent protection. One of the great benefits of an immunocontraceptive is its lack of endocrine or metabolic side effects, which makes people more prone to take it. It doesn't require insertion or an implantation of a device. The way an immunocontraceptive works is it develops antibodies that bind to the targeted protein and block the function. Antibodies are proteins that bind very tightly to their antigens. They are produced as a defense against infection. When foreign material enters the cell, specialized cells known as lymph stems recognize it as alien. The recognition of the foreign material and antigen triggers a chain reaction in the immune responses. Cells in the lymph system, known as type B lymphocytes, produce an antibody made to match the particular antigen. Now, for the immune system to recognize its own body's material as foreign, a foreign substance must bind to the cell in question. This tricks the body into thinking its own cell is an antigen. The duration of the contraceptive would depend on how long the antigen could remain bound to the cell, could remain bound to the antibodies. Essentially, immunocontraceptives require the production of a controlled, time-limited, and non-pathogenic immune response to components of the reproductive process. There are several studies possibly going on that use the CATSPER1, and they're underway at institutions all over the country. Immunocontraceptive development is still in its very early stages and needs to be proven effective, safe, and reversible before it can be marketed. However, the fact that the CATSPER1 gene has been identified as negatively affecting sperm motility means scientists are well on their way to discovering how it could be used as a male immunocontraceptive.